Thank you. I would like to take you into a small tour, 20 minutes tour, on short proteins. But before starting, let me tell you a little bit what is known about short proteins. So short protein, I would like to claim that short proteins are actually an unexplored niche of the eukaryotic proteome. And why is that? From the genomic point of view, it's very hard to get them, mostly because the ab initio gene finding methods basically fail in finding them. It's true also for NGS and so on. From the proteomics point of view, it's even worse because there are a lot of modifications that cause missed spectra, but even worse, if anyway they were not in the database, it's of course impossible to find them in the database. So it's a little bit of a circular argument why they are so hard to get. But in addition, there is another pretty surprising observation that I want to share with you, that whenever you have them, they are mostly uncharacterized. What I mean by that, if you just take the entire Swiss prot that's supposed to be highly annotated, and you take the short proteins, never mind what's the cutoff, you can see quite nicely that there is an inverse relationship between the length and the characterization. So the shorter the proteins, the higher the fraction of the uncharacterized one. So not only that it's hard to get them, once you get them, you don't know what to do with them. So this is really setting the stage why we need maybe some other way to get to their functions. So one exception are short proteins that belong to animal toxins. And the way maybe to show it is if, again, we take the Swiss prot, we take all proteins that are below 100 amino acids, and you look at their annotation, you can see that toxin, neurotoxin, and specifically ion channel inhibitors are extremely short. You can see the p-value that most of them are really below this 100, which means that they are very enriched. Okay, and so let's say a few words about these ion channel inhibitors. What you see here in this picture are two of those coming from this conus on the picture. They look quite the same. The sequence is, their sequence are extremely short. And if you look, I won't let you too much time to look at it, but as you can see, the similarity on the sequence base is extremely embarrassingly low. And actually, those two are acting on a very, very critical receptors or channels in our body. Basically, they will block transmission of neurons, so they will cause, I don't know, the elephant to die just because of this small peptide. So it's really very potent on one hand, but sequence doesn't tell us much. So there is minimal sequence similarity in between. What about to do it from the structural point of view? So indeed, it is known that they are shared, all those ICI or ion channel inhibitors shares many, maybe 100 superfamily and somewhat more of that. However, there is something very puzzling again from the structural point of view. There are, in the left, you can see that there are many folds that will hit one target. In this case, potassium channel. But on the right side, you can see that you have the opposite situation in which one fold from this scorpion will hit many, many targets. So all this nice connection between targets and folds is a little bit used differently in this case. So no unified structural story. So the question is, what makes them characteristic? What's the key? And the answer is pretty simple. It's a common feature of stability, of compactness, that is based on a link of disulfide bond that really capture the scaffold of those very short proteins. So exactly what I told you now became a basis for developing a feature-based predictor that was done many years ago by Noam Kaplan in our lab when he was a student. The idea, like in many machine learning predictor, is very simple. You take what you know, which is a few hundreds of the positive sets, many negative sets. There are a lot of details here, but you use it to learn on new observation that you haven't seen before. And I just want to encourage you to come to one of the posters in which the most important part, which I won't talk about, is how to really represent those features. This is the most interesting and I would say computational, also challenging. once you do this, the rest is pretty much straightforward. So I'll skip this. And the goal, as I said, is to find what we call TOLIPs. TOLIPs are toxin-like proteins, as abbreviation for those compact structure that we know nothing about. Okay. So we developed this machine that we called ClanTox for classifier of animal toxin protein. And you can see in this scheme a result of one test in which you take all short proteins in the Swiss prot, and you see that the big, hill there are all the negative results, and on the really tip of the here you can see a tail that associated with highly predicted. We call them candidates because we don't know yet, but they are TOLIP candidates. So take-home message number one is that the ClanTox output is a list of short proteins with TOLIP properties, the way I defined it. So the highlight talk is based on a research done by Yitzhak, Itai, and Manor, really to understand toxin-like sequences in a very restricted part of the evolutionary tree, much before the bilateral symmetry has been evolved. So in a bit weird kind of branch. Why do we care? So why do we care about this marine animal, very strange one? So one argument is that those are among the most poisonous animal on Earth. This is from the Guinness Book of, number one being very poisonous. More importantly, from the biological point of view, they have a huge spectrum of activity towards receptor, molecules, transporter, and a lot of membrane proteins. And what I want to emphasize that they have a most extreme evolution creativity, and I'll specify what I mean by that. Just some facts. If you go to those conus that, as you can see, were found in Uruk 5,000 years ago, from 5,000 years ago, each of those species produce over 1,000 of bioactive peptide. Unbelievable. Now, those have a high potency, but very broad specificity toward any almost surface protein that we know of. So many of them went into the clinic as drugs already. Now, let's go to this Cnidaria genomes, and when you look at the two examples that were fully sequenced, one of them is the Hydra and the other is the Nematostella, the sea anemone, you can see quite clearly, look at the blue color that are annotated part, that the annotated part is really very low. Namely, those genomes are poorly annotated in general. However, as I said, and I want to show the procedure that one can do to take the genome, take what seems to be short proteins, feed it to the ClanTox. I already explained the ClanTox pathway. Then get the prediction, take the candidate TOLIPs, and do some functional assay to understand what they're doing. So this is the procedure I'm going to go with you. So once we did this and we put it in the ClanTox, we found quite surprising facts. In the Nematostella, we found 14%, which is huge, of positive, very highly positive prediction. Actually, in the Hydra and in other organisms such as sponge, you don't find it. So among Cnidaria, some display a very rich repertoire of creative solution. I'll just take an example. Those are a list of toxins, and as you can see in this top panel, you can see that there is a huge expansion of those toxins in this specific Nematostella. Let me take you to a sidewalk just for 1 minute to show you that the creativity is more than what is in several aspects. For example, what you see here is the output of ClanTox. Each blue line is a protein according to its length, and in the red one you can see the cysteine position. So the non-toxins are a subgroup of this, and most of the other were initially unknown, but with some bioinformatics data mining, you can see that they're related to some quite interesting function, such as antimicrobial, calcium binding effect, protease inhibitors, and so on and so forth. Altogether, we found 80 new interesting candidates. Now, the Nematostella has another quite surprising and unexpected feature showing this creativity, which they have a huge amount of tandem repeats. If you can see this list, you can see that all other genomes have anything between 5, 7% to 10%, while the Nematostella have almost 20% of tandem repeats. I'll keep it just as a showing that these genomes are totally crazy. Now, after I told you everything that I wanted to tell you about the oldest metazoa, let's go to some more modern metazoa, such as human. So the idea was, if this is true, why should this creative idea be restricted only to Cnidaria? Why not human? Why not mouse? We said, let's check it. We went to the mouse genome from deep sequencing. We took everything that are short sequences. We did some filtration that I don't bother, and we found 40 cases of very highly selective TOLIP-like. So what is it? So the surprising story is that among mammals, we found unexpected richness of TOLIPs. So I'll show you one group of those TOLIPs. On the top panel, you see a 1.1 million bases in our chromosome, in the mouse chromosome, sorry, and a list of many predicted and unknown genes, 19 in altogether. 5 of them, we call them as a new entity, ANLP, for alpha-neurotoxin-like proteins. And when we went, just to show you again, when we take all 5 of them and align them, you can see how bad, how poor the sequence alignment among themselves. I mean, it's really embarrassing, despite the fact that the structure model is quite stable, and it's belonged to a very known family of 3-finger fold. Now, we take this protein. Burkett says that I used to be a biologist. I am still a biologist. We went to the lab. We expressed the protein in macrophage, and we did quite a bit of biochemistry, and to our delight, we found something quite interesting, that this ANLP, unknown TOLIP from mouse, act as an inhibitor, modulator of acetylcholine receptor, one of the major receptors in many biological functions. Now, actually, it's defined the inflammatory response of those macrophages, and I'll go briefly just to show you that you see those dots. You don't have to understand too much, but just to know that this is a panel of cytokine that are released by the macrophage. If you do this and you put in the ANLP3, you can see that you get a new set of cytokine, namely that this addition of these very short peptides completely block the effect of nicotine. So before there was a nicotine block, it's gone, which is very nice modulatory function. So what are those TOLIPs? So just to show you, they look like this, and they belong to a very big clan. This is a PFAM clan in which I talked about the toxin. There are other subfamily that I don't have time to talk about, but the biggest one called Li6, UPAR Li6. And the ANLP belongs very nicely to this one. So what are those Li6? Li6 are 30 genes, again, so it's a little bit like a treasure hunt now. 30 genes in mouse, they look like this. They have this fold that I showed you. They were discovered 35 years ago with no information about their function, basically, but it is used as a surface marker for all kind of function definition. So this is from a review from last year. But again, all those 30 genes are also organized in a very nice cluster at the chromosome. It's another chromosome from the one that I showed you before. So you can see the human and mouse together. You can see that there is a very nice match of human and mouse, but an addition of expansion in the mouse. This is the mark of the addition blue, red genes that are mouse related. So it's a very, very extended family, as I showed you before in the Cnidaria. So just to finish, I want to show you that the evolution innovation comes in two flavors, on the immunological and neuronal aspect functions. And the Ly6 cluster, I mentioned it, but I want to show you something very interesting. They have many Ly6. However, they built a code to define the different cell type of the immune system. So the immune system, it is known that they have many cell types, the B, the T, the NK, the monocytes, and so on. Each of them has its own pattern of LC variants. So this is quite interesting. Is it only on the immunological aspect? The answer is not. Because if you go to the brain, and this is specifically going deep into the hippocampus, the learning, memory, and plasticity region, and you look at two of those representative in this chromosomal section that I showed you. One is called Ly6, the other is important in prion disease. You can see that by in situ hybridization, each of them have its own expression. So what you see in red is the high expression of specific genes. So you can see that they are almost complementary view, each to another. So each of them bind or expressed in a specific subset in the hippocampus. So keep in mind that TOLIPs, I'm putting it as a statement, they can code for cell identity in plasticity-dependent manner, such as the immune system and the brain. So just before finishing, I want to show you that Ly6 are also involved deeply in a lot of diseases, systemic diseases, such as cancer, psoriasis, myelination deficits, schizophrenia, Alzheimer, and so on, atrophy of muscle. And to show you what I show you in this treasure hunt, we started with the toxin, right, like the Bungar toxin. We show you that there are many more to this family, the Ly6, the ANLP, and so on. They all look quite the same, but look at where they are involved in disease, in pain, in immune system, in Alzheimer, despite the fact that they look the same. So TOLIPs leads to a range of systemic diseases and functions. To summarize, TOLIP in Metazoa, I showed you about 80 or 100 TOLIPs coming from this creature. Many of them are real toxin. I'll show you about 100 or 40 in mouse that have no toxin in the classical toxin type of a definition. However, they both target receptor. They both are very short. They have a lot of modification I didn't talk about. They both use chromosomal amplification as a mode of expansion. However, on the left side, those are irreversible function. When ion channel, one of those toxin, hit a channel, it's block it. That's it, one-way ticket. Those modulators in the human and mouse are reversible. They can come in and out. And that's make a huge difference because they can have a modulatory action, cell recognition, adhesion, cell lineage, and immunological code, as I showed you. So lower affinity, broader specificity. Do they have more creative ideas? I'll show you two. One is they have a lot of alternative splice. Some are with GPI, some are without this anchor. As you can see, short deletion makes them new variants. But what about the other? Do they have a creative protein-protein interaction? We just heard about how you build up protein-protein interaction. So let me show you an example. In the A part, or let's start with the B. Those are homodimer in which they have a very nice, well-defined interface between the two subunits. Look at the left side. Those are two heterodimers, but their connection is through a cysteine bridge that jump from one to another. So they have another creative idea to put them together. So this is just to show how diverse is the family in solution. So that's exactly the message. Being short allows you to use multiple solutions for gaining diversity. So it's a big statement, but I think I showed you a few examples to support it. So, summary. I showed you a methodology for identifying bioactive short proteins. I showed you how cell surface reversible modulation can be part of our own kind of regulatory mechanism that was a little bit hidden up to now. I showed you some on the evolutionary insights, such as expansion, invention. I didn't talk about, but many of them are candidates for therapeutic toolbox, I would say, for the future, and they act on the brain, immune system, and cell-cell interactions. So the hypothesis that I put in the first slide told us that short proteins might be unexplored regulatory modes. Or is it? I think yes. Is it a playground of evolution? I think yes. And they are key modulator associated with disease. I think I showed you a little bit of each. So thank you very much, and I'll be happy to take questions. Thank you very much.
My question is, because they are hard to annotate,
yes.
Do you explore, predict, taking the previously annotated proteins,
no.
Or do you do DNA scanning?
So it's a very, very good question. Actually, on purpose, we do completely nothing, no data. So it's only FASTA file that gets into the machine. And we have later on few filters that we can use to sort out the result. So one of the way is, for example, to say if, but only now, if we see signal peptides, which is the hallmark for being secreted, that will be a good sign that we didn't just catch a pseudogene or something. So we have other filters that come later, but never in the predicting stage. And in this poster, we discuss it also quite deeply. No, the domain is beautifully characterized. That's the beauty of it. If you go to the L6 recent extensive review published last month, you can see the following. You can see that this is their review. I didn't put the author, but it's from last month. You can see that they are really big family, but their character, this is the summary of the review. They are large family, 35 years ago, by chance, isolated. There are surface markers, they are used for sorting cells. So they are used heavily in the clinic to say this is that. But all the rest, like are those important in neutrophil biology maturation? Is it cell-cell recognition? Completely unknown. But I gave you a few hints to show that they might be a binder to key elements, such as the ANLP that I showed you specifically, that it's bind to a specific receptor, and they are from the same family. So I'm sure that either they bind to integrin, to one of those. I mean, we can talk about more biology, but they belong to this super binders, low affinity, but a broad specificity group with very little information.
I was wondering if you have used these TOLIPs as markers of tissue evolution, because such as you are showing, they seem to be able to distinguish between different tissues or structures in the brain. Yeah. So, with respect to find a specific expansion associated with this.
Yeah. So the evolution. First of all, we didn't go into learning the evolution, in the style that you were mentioning. I mean, looking what's conserved, synonymous, non-synonymous, and so on. What we could do quite easily is to show where the duplication, the local duplication happened. So you can draw a very nice story. It started from this guy, it's expanded to this and this. So you see so for example, in this ANLP, there are 9 in mice, but only 4 in humans. So there was less one duplication. So it's very easy to follow the history. And the trick, I think, because they are so short, but there are, by the way, 3 exons. It's not like there are 1 exon. It's a real gene in a sense. They are very fast going. So, and I think it's the rule for short proteins, but I didn't show evidence for that.
Any other question? Then thanks again. Oh.
Do you know whether these polyps are outside metazoa, like protozoa or bacteria?
Yeah. The largest group of polyps, we have few papers on polyps in viruses. It's crazy. Viruses grab them from their host because they are so cool. So in viruses, they are not in bacteria, but there are a huge amount of them in plants. We don't understand exactly why. Maybe because the plants had to replace their brain by other clever communication methods. I'm serious. So there are a lot of them in plants, but they go all the way to sponge, but not before.
Thank you.